We all strive to live a long, prosperous, and healthy life. With advances in health and medical sciences, this goal is ever more attainable. The Sam and Rose Stein Institute for Research on Aging is a nonprofit organized research unit under the auspices of the University of California at San Diego, committed to advancing lifelong health and independence through research, education, and patient care. To better empower and improve the lives of young and old alike, the Stein Institute presents the following program.
What I'm going to start with is to give you an idea how people thought about the human brain's structure at the time that these techniques became available, at the time that I entered the field. There was a sort of a dogma at that time. It was kind of built up mostly from observations from autopsy material and to some extent from earlier methods for visualizing the brain in much less detail. But this was the old view, and a view which I think is surprisingly still encountered quite frequently out even in the scientific field, about the structure of the human brain. This is the old view. First of all, we were taught as graduate students that all of the neurons that are present in the brain are there at birth. So, and furthermore, they're pretty much where they are going to end up at birth. And that after that point, neurons are just lost in the brain. Now, we all know that this first tenet of the old view or the old dogma has fallen to basic neurobiology. And we now know that although it's true that the great majority of neurons are born before a child is born in the human brain, there are limited areas in the brain where there is some continuing neurogenesis that goes on throughout life. So already we know that this original first tenet didn't quite hold up. But it was appreciated at that time even that there was a very exuberant process just after birth in early postnatal life of what's called myelination of fibers in the brain. And what I mean by this is a sort of deposition of a lipid in the axons of the brain. These are the cables that connect the neurons in different parts of the brain. So it was appreciated that there was this biological development, ongoing biological development of the brain's fiber tracts that occurred rapidly over the first few years of life. But we were taught that the research to that point had suggested that the brain structure was essentially adult-like by the time a child reached the age of about 5, and that certainly the growth of the brain appeared to be complete. What this meant was that we expected that brain morphology, or basically the hardware of the brain, was in place and pretty stable across childhood into late childhood and adolescence and through most of adulthood. But people were well aware that at some point there seemed to be some kind of regressive changes that set in, and it was expected that these changes would begin sometime after the age of 60. And this was very much the focus of the field of human brain research at the time that in vivo brain imaging techniques became available. And so this is just an example of a set of magnetic resonance images, and it shows you how much more detail we were able to appreciate about the human brain once these techniques became available. And we were really able to examine these tenets of the dogma about human brain structure for the first time in living individuals and in large samples of healthy living individuals across the age range. Now, it turns out that although you obviously can see a lot of detail in these images, they're really remarkable. And these are just different types of MR images that show you different kinds of biological characteristics of the tissues. Nevertheless, there's a lot of individual difference variability in the size and shape of the human brain, just like there's a lot of difference between us in the sizes and shapes of our bodies and our faces and our noses and everything else. And so just to look at the images, it wasn't absolutely obvious how much they were changing across the age range. And so it remained for us to develop a set of techniques that are now collectively referred to as computational morphometry or MR morphometry methods, for us to begin to really systematically examine anatomical or morphological properties of the brain across the age range quantitatively. And there are a number of these techniques that have developed, and in fact, large schools of techniques that have developed. And it would take me all of this talk and many other talks to describe them all and their advantages and disadvantages, and I'm not going to do that. But this is just to illustrate that these images, these MR images that we were able to get, have to be further processed before we can really quantify and measure morphological characteristics. And this is just an example of some images that were processed in my laboratory, and I'm going to show you a fair amount of data using these methods, so this gives you some idea where they came from. But you can see that we have to separate the parts of the brain, the parts of the images that are the brain from the parts that are outside the brain, and the skull and the eyes and things like that. And we have to do some further computation to try to identify the major tissues of the brain. And in these images, I've tried to color code the white matter and the gray matter of the cortex, and the gray matter of the deep subcortical structures. So we separate those structures so we can look at individual deep subcortical gray matter structures and individual lobes, they're color-coded here too, of the cerebral cortex, the underlying white matter of the different lobes, and so on. So that virtually all of these morphometry techniques essentially move in this direction to some extent. They try to identify the different tissues and then to define the boundaries of different brain structures so you can sort of begin to look in detail at the brain structure. And this is another technique that's used in a laboratory with which I collaborate closely. This is from another UCSD laboratory of Dr. Honors Dale. This is a technique called free surfer, and you can see that the methods have developed to provide even more precision in the different regions of the cortex. So here you see much finer detail in the cerebral cortex, and these methods are available now. Okay, now let's revisit this conventional view that existed when the techniques became available. What we were expecting was that adult brain structure would be present already in school-aged children, and that brain morphological characteristics, or again, the hardware of the brain, would be pretty stable across childhood and adolescence and the adult years, and that we would at some point begin to see some evidence of atrophy. And really, our early studies were pretty much focused on this question: when would we begin to see age-related atrophy in the brain? And so what I'm going to show you now is some very early data that we had, and these happen to be data from my laboratory, but where we're just estimating the total cerebral gray matter volume. And we just adjust it for head size, because people with big bodies and heads have bigger brains than people with smaller bodies and heads. So we just adjust it for that. So you're just going to get an estimate of the total gray matter volume in the brain. And again, this is what we were expecting to see, is a function like this. And we were really doing these studies at the time because we were interested in when this began to happen and how frequently it happened in normally developing or normally aging people. And this is what we actually saw. So first of all, not only was there never a time across the lifespan, the age span, when there was stable gray matter volume, it was the case that actually the most dramatic age-related changes occurred in the earliest part of the age range. I think you can see here that these changes across the age range are even more dramatic than they were later in the age range. So this was a great surprise to us. This was completely unexpected. And these observations that we made very early here at UCSD, they were just sort of among the first that were made of this phenomenon, really tended to refocus the field on this earlier part of the age range, as you can imagine, and really got us thinking about the assumptions that we started these studies with. And I'm going to take a few minutes to talk about this this earlier part of the age range, because I think really to understand aging, you really need to understand what's happening all the way across the age span. So let me talk a little bit more about this focus that was stimulated by these early observations on brain maturation or biological development of the brain. Now, since those early observations that we made in our lab here at UCSD and in several other labs, these very advanced computational morphometry techniques have been applied, and a number of additional studies have been done. And this is a summary slide from all of our favorite study, which is done at the National Institute of Mental Health in the laboratory of Dr. Jay Geed. And this is a graphic that shows you a summary slide of changes across this age range from 5 to 20 of the cortex, of the cerebral cortex. And here, blue colors represent thinner cortex, okay? And what you see here is that if starting at age 5, the thinnest cortex is just in what we refer to as the sensory motor cortex. These are basically the cortices that are most involved in just your basic sensory and motor processes. And that this thinning spreads out gradually across the age range into what we think of as the secondary and tertiary cortical areas, away from those primary sensory motor areas in the brain. And you can see that there's this ongoing thinning up till age 20 here. But you kind of get a hint, even in this picture, which shows these rather dramatic thinning phenomena in the cerebral cortex, that even at age 20, it might not be entirely finished. And these are just some data that were collected in Elizabeth Soul's dissertation, which was done in my laboratory. And these are some analyses that were done when she went as a postdoctoral fellow to UCLA and joined Art Toga's lab. But you can see here that this is just another way of looking at some of these same changes. These are differences in the cortex in the posterior frontal lobe and in the parietal lobe. In children, differences between groups of children aged about 8 and adolescents aged about 14. But what was really striking here were the differences that we saw in the 14-year-olds relative to young adults who were about 26 years of age. We saw continued dramatic changes in the cerebral cortex, shown here in these little purple blobs, in the frontal lobes, but also in this green area here, which represents deep gray matter structures deep inside the brain. And I'm going to zoom in on those for a minute and show you the data. This is the thalamus, and you see the changes here early in the age range, in adolescence and early adulthood, and in the nucleus accumbens. Now, the thalamus is a very important, some people would say, to oversimplify a little, a kind of relay station. Virtually all the information going to the cortex, it goes through the thalamus, and the thalamus is very important in processing information that's going to and from the cortex. So an important processing center deep in the brain. And the nucleus accumbens is a part of a set of structures we refer to as basal ganglia. And this particular structure has been strongly implicated in social and emotional functions, has a lot to do with our sense of reward or satisfaction, has to do with craving in some cases. So this is an important structure in the motivational system, if you will. And clearly, there are some fairly dramatic biological changes occurring in this structure into adolescence and early adulthood. Okay, now here, most of the data that I've showed you so far, I think all the data I've shown you so far have been they're cross-sectional studies. But this is a study, these are results that are taken from, again, Elizabeth Soul, who was a graduate student here and is now a professor at UCLA, and a study she did with kids who she scanned at age 7 and then again at age 9. So these are longitudinal studies. Each child was studied twice at 7 and 9. And what she saw was very similar to what I've shown you generally, the evidence of widespread thinning of the cortex over that 2-year period. But the interesting thing here was that she also saw that in some very specific areas around the temporal lobe, particularly on the left side, local areas of thickening of the cortex over this age range. So now I think already you've gotten the idea that the cortex, if you just look at something like cortical thickness or volume of the cortex, is a fairly dynamic thing, especially early in life. But I've told you a lot about apparent thinning of the cortex, loss of volume in the gray matter structures. So you might be wondering, why don't these kids' brains look kind of atrophied and shriveled up? I mean, they're losing all this gray matter. Why don't they look atrophied? And the reason, of course, is because concurrently, at the same time that these changes and these apparent cortical thinning changes and these apparent losses of volume, gray matter volume in the deep subcortical structures, at the same time this is happening, there is this opposing change, a progressive change in the cerebral white matter. As you see, this is just to remind you that that's this white stuff here underlying the cortex. And this is just some data from our lab again, showing estimates of the volume of the cerebral white matter over the same age range that we've been focusing on. And you can see that these progressive changes are offsetting these regressive changes happening in the gray matter. And this, you see, is why people thought there was no further growth, because there were superimposed progressive and regressive changes that were going on throughout childhood and adolescence, but the volume of the brain wasn't at some point growing anymore. And that's one reason there was this misconception that brain growth was complete at age 5. And this increase in volume is, of course, the ongoing process that we knew about, which is this proliferation of oligodendrocytes, a glial cell class in the brain, and the deposition of these myelin sheaths that get wrapped around the axons that are connecting the neurons in different parts of the brain. So this myelin deposition is increasing the volume of the cerebral white matter. Now, this is an important change in the structure of the brain, because we know that the purpose of this myelination is sort of similar to the purpose of insulation in electrical wires. It improves the transmission of electrical currents down the axon, and because of the structure of it, it results in a much more efficient transfer of electrical signals from one part of the brain to the next. So it has important functional consequences. At this point, we certainly were aware that there were clearly important dynamic ongoing changes in the biology of the brain. And we could see that there were white matter volume increases that were offsetting what appeared to be gray matter volume decreases in the brain. But with conventional MRI, we couldn't really take the research much further. We were very interested in the changes in the sort of axon, the fiber tracts, the brain's fiber tracts, but we couldn't really see the structure of the fiber tracts very well. Fortunately, a new technique came along with MRI, which is referred to as diffusion tensor imaging, and this really changed that situation and opened up a new window on the development of the brain that's been very important, and I'm going to tell you a little bit about that now. Now these are conventional images. I've been showing them to you. This is what's called a T1 weighted image and a T2 weighted image, and you can see that you don't get much detail about the structure of the white matter. You can see where it is, but you can't see much detail about its structure. This is a diffusion tensor image which codes for a measure called fractional anisotropy, and I'm going to talk more about that in a minute. But I think you can see that the signal is highest in this image in the white matter of the brain, in the fiber tracts of the brain. And this image is a diffusion tensor image which has been further coded so that you can see that not only are we able to identify the fiber tracts in the brain, but by manipulating this information from diffusion tensor imaging, we can also get some information about the direction in which the fiber tracts are traveling, so to speak, the direction in which they're coursing. So this image is color-coded so that you can see that the green tracks are tracks that are coursing front to back through the image. The red areas are tracks that are coursing side to side in the image. And the blue areas, if you can see them, are tracks that are coursing through the image, from above to below the image. Okay. Now, what is diffusion tensor imaging? It's a method for measuring the diffusion or motion of protons that are in the water molecules in the brain. And what's interesting is that you can not only measure the amount of diffusion, but the direction of proton motion within each of these little voxels or little chunks of tissue that you measure with this method of imaging. So you can describe the direction of proton motion with a mathematical construct which is referred to as a tensor. And I'm going to try to give you some idea what tensors are in a minute. Now, this is interesting in part because proton diffusion that's measured with this method tends to be relatively isotropic. That's a term that we use with this method a lot. This means that the motion is kind of directionally random. The motion is sort of equal in all directions. That's what we call isotropic. And it tends to be isotropic in gray matter. But because the fiber tracks have this linear structure, they're basically bundles of cables, if you will, this linear structure tends to constrain the motion in such a way that the proton diffusion is disproportionately along the long axis of those fiber bundles relative to the orthogonal or independent or perpendicular direction. And so this produces what is referred to as anisotropy. Now, let me talk a little bit more about that. I've taken this slide, borrowed this slide from a colleague, Guido Gehrig, because it has a nice visualization of these tensors. And you can see here that the tensors are really measures that allow you to characterize the diffusion in three cardinal directions. And so in this case, this is meant to depict diffusion which is isotropic, that is to say, the three vectors are equal in length. And the size of the diffusion tensor is a measure of the total amount of diffusion that is measured in the voxel. Some voxels, however, will exhibit a diffusion which appears to be primarily along one axis relative to the others. So by computing what's called the principal eigenvalue of the tensor, you can see that there is more diffusion along one direction than there is in the other directions. And so the tensor has a kind of football shape or an elongated shape. And the measure of the elongation of the tensor is often referred to as fractional anisotropy, or FA for short. Let's look at some of these brain images. This is a measure of the size of the tensor, the amount of diffusion that's present in each position in the brain. And you can see that in these fluid-filled spaces in the middle of the brain, there are larger diffusion tensors. There's more diffusion because the water and the protons in the water are free to just diffuse freely, and so they diffuse a lot. But when in the brain tissues, where there are a lot of cellular structure, a lot of the water molecules are bound to the cellular structures, then the diffusion is much less. So the tensor is smaller. So this is just a map of how much diffusion is present in each position. And you can see that it's good at telling you where the fluid is, but you don't see much structure in the white matter, because all the brain's tissues show lower diffusivity. But if you look at fractional anisotropy, that is the degree to which the tensor is elongated, then you see quite a lot of variability in the white matter here. If you think of a lasagna pan, and it's filled with water, and you take a food coloring dropper, and you drop a drop of food color into your lasagna pan of water, what you would expect to see is the color would spread out rapidly in all directions. If instead of having water in the lasagna pan, you had a rectangular sponge which was very wet, and you dropped the color onto the wet sponge, it would also spread out in all directions, but it would be slower. So that's sort of the difference that you're seeing illustrated here in this diffusivity image. Okay. But now imagine that in the lasagna pan you had cooked spaghetti, which was very wet. So there was just water in it up to the top of the spaghetti in the lasagna pan. And all the spaghetti was lined up parallel to in the Lasagna all lined up straight. Then you can imagine that if you dropped the food coloring drop in, you would see that the food coloring would sort of spread out along in between the spaghetti noodles pretty quickly, but it would have take a little longer to get sideways through the spaghetti noodles, if you see what I mean. And that's kind of the difference in diffusion that you're measuring with this fractional anisotropy measure. And that's what we can exploit to look at the microstructure of the fiber tracts in the brain. So now, what happens with age to these parameters from DTI? Well, this is a model that was developed for explaining the developmental changes, but I think they're useful for understanding how age affects the DTI parameters generally. This is supposed to be a depiction of the sort of a cross-section of a fiber tract bundle over here that's immature, and this is a cross-section of a more mature fiber bundle. So the circles here, the larger circles, are the axons that you're cutting through. But in the immature state, there's a lot of water, free water, around the axons. And those little dots are the free water molecules around the water. Those little dots of water molecules in the free water around the axons, they can diffuse pretty freely, and they produce a lot of diffusion. And they produce diffusion in all directions equally because they're not too much constrained directionally. And these are supposed to be the estimates of what the three eigenvalues would be. This is the largest one, the diffusion along the long axis of the fiber tract, and these are the diffusion along the other two orthogonal axes, okay, in the immature state. Now, as myelination occurs, and for other reasons perhaps, as the fiber tract becomes more mature, the axons become thicker, perhaps because they actually increase in diameter or caliber, or perhaps because they become larger because of the myelin sheath being wrapped around them. And there's less of this water space around them. In other words, they're more densely packed. There's less water there. And because of the loss of these free water molecules from the tissue, all three of the diffusivities go down. There's less diffusion overall. And they all go down. This is sort of isotropic diffusion. This is the randomly directional diffusion. It goes down. But because there's less loss of diffusion along the long axis, perhaps as you can see depicted here because of increased axoplasmic flow in these larger axons, this diffusivity estimate goes down less than these do. And because fractional anisotropy is the proportion of the total diffusivity that's in the first eigenvalue, that means that this fractional anisotropy goes up. And that's what we see in development. These are actually data from Christian Boyot's laboratory in Canada, just showing that if you look at this principal eigenvalue, this one that's along the long axis of the fiber tracts, in two different fiber tracts, there is a reduction, but it's not as steep. I think that's what this is meant to depict. It's not as steep as a reduction in the other two directions, which is consistent with this model about what's going on in development. And that's true both places. And these are some data from my lab. This is in children 7 to 13 years of age. Again, this is the principal eigenvalue, what's called parallel diffusivity. It goes down, but not very dramatically. This one goes down a little bit more dramatically, and because FA is the proportion that this represents of the total, fractional anisotropy goes up. So this shows the increase in fractional anisotropy with age. Now, because you also get information about the direction, the actual direction of diffusion in each tensor, this information can be further manipulated through techniques referred to as streamlining or track tracing or tractography, and these are still rapidly developing methods, but so far the results have been quite promising that you can use these data to identify specific major fiber tracts in the brain that are known from previous anatomical studies to be present in the brain, You can find their locations and identify them in the brain, and then you can measure these diffusion parameters in them, in each fiber tract in the brain, and look at how that changes across the age range. And this is, again, some beautiful figures and data from Christian Boyau's program. And he's looked at kids and young adults, and he's shown that some of the major fiber tracts in the brain show quite dramatic, quite early biological development, reaching or approximating adult levels of FA about age 11. But look here how long after, in this postnatal period, that everything was supposed to be happening in the first few postnatal years, look that even in these fairly rapidly developing tracts, it takes until about age 11 for them to asymptote at adult levels. This, you can see, looks pretty stable out here to about 35. Some tracts show a more protracted course of development, not reaching or approximating adult values until age 15 or 17. And a few, interestingly, show an even more protracted course of biological development and don't approach adult levels until sometime after age 20. So really this was, again, quite an eye-opener and really gives us a window into the ongoing biological development of connectivity in the brain. Now I'm going to shift, and we're going to look at brain aging on the other side of the age range. And first what I want to do is just show you visually what some of the changes look like in brain aging that were pretty much accessible to us early on, as soon as we had an opportunity to look at brain imaging, MRI, structural brain imaging in people of different ages. And these are just some images. This is an image, a typical image from 32, a couple of images from a 32-year-old and two 80-year-olds. One thing you notice is different about both of these 80-year-olds from this 32-year-old is this increase in this high signal area around the edge of the brain. Now that's cerebrospinal fluid. That's an increase in the fluid that's surrounding the cerebral cortex and also in the deep ventricular structures in the brain. These are the fluid-filled spaces of the brain. And this increase occurs because of loss of tissue volume, of brain volume, in these individuals at some point across their age range. And this is, as you can see here, as illustrated here, this is a very common finding. Virtually all of us, by the time we reach 80, show some sign of brain volume loss in the brain tissues in the brain, as indexed by this increase in the cerebrospinal fluid in the brain. Now, another thing that you can frequently see in older individuals is changes in the signal values in the underlying white matter. Again, these axons, these cables that connect different parts of the brain. And here you can see, may be hard to see if you're not used to looking at these, that there's, in this individual in particular, these bright areas in the white matter that are not present over here in the young individual, nor in this 81-year-old. I've sort of processed the brains down here as color-coded things so you can see them more easily. They are these red areas here. And unlike this volume loss, which we see so frequently in older individuals, this kind of change is not at all universal in older individuals. And so here you can see this 81-year-old shows very little of that kind of change, and this 80-year-old shows a lot of it, actually, more than most individuals show. And so these are the kinds of changes that we wanted to characterize with MRI when we started doing this research. Now, to date, there have been a lot of studies looking at the sort of average changes across the age range in the thickness and volume of the cerebral cortex. And they pretty much all agree at this point on some basic facts about the average change across the age range. Focusing now on the adult age range, this is just an estimate of the frontal cortex. That's this part here in purple. And many studies have now shown that this change, this basic function that describes sort of thinning or loss of volume in the cerebral cortex is pretty similar in different parts of the cortex, but it seems to be a little bit more pronounced in the frontal lobes than in other lobes in the brain. However, not all of the gray matter structures in the brain show the same kind of function of volume loss across the age range. The hippocampus, for example, which of course is a structure of great interest to people who study aging and dementing disorders because it has been so closely linked to memory functions, shows quite a different function, so that it appears that the hippocampus actually continues to increase in volume through childhood and into early adulthood, and only shows a loss of volume later in life. So there is this curvilinear function, and it's not well understood yet why the gray matter of the hippocampus shows a different age function than the cerebral cortex, but clearly it is different. And in the beginning it was quite difficult. The findings were seemed to be contradictory because people were studying different parts of the age range, and of course, since it's curvilinear, the effect that you get is very different depending on which part of this curve you look at. So, now what about the cerebral white matter? Well, I showed you previously that there are quite protracted increases in the volume of the white matter into early adulthood, and that continues, and actually the peak probably is somewhere around 50. So there's a very protracted increase in the volume of cerebral white matter, and then gradually you begin to see volume loss out in the 70s and 80s and so on. Now, what about this signal alteration in the white matter? If you measure the amount of signal alteration in the white matter, which generally signals an increase in the water content of the tissue, probably because of loss of structure or loss of myelin or some kind of damage in the white matter structure, and it can occur for a number of reasons, so it's a little bit nonspecific. But if you measure the amount of it, this is what you see in normal individuals. And what I want to draw your attention to is the fact that it does become more and more common in older individuals, but it is highly variable in older individuals, with some older individuals showing essentially none of this change and other individuals showing quite a lot. So it's a very variable phenomenon in brain aging. Now, what is this new technique, diffusion tensor imaging? What has it added to to the picture about brain aging and changes in brain structure? The findings with this technique are quite parallel to what we saw with brain volume. We see that mean diffusivity, or the size of the tensor, goes up, and fractional anisotropy goes down. One thing that I think we have to really notice with all of these distributions is a tremendous amount of individual difference variability. And so now I'm going to talk about how we interpret individual differences on these measures, because I think this is where most of the interesting recent findings are. Well, with the children, in the early days, we noticed here in this early age range, this is just kids 6, 7, 8, 9 years of age, a tremendous amount of difference in the frontal lobe volumes of these kids in this first study that we did of developing kids. And we were interested in this group of kids who up to 16 years of age, what this kind of variability meant. And in Elizabeth Sal's dissertation, we were interested in the development of memory functions. And in that first study, we were able to show that the more adult-like, that is, the lower the cortical volume was, frontal cortical volume was, the more adult-like the cortex, the more efficient the child's memory retrieval abilities were. So in this case, we interpreted that as meaning that a child who was more advanced in terms of the biological development of the frontal lobe showed more adult-like memory functions. And later, Elizabeth did another study, which was kind of consistent with this, which showed that if you took these kids that I told you that she studied who were 7 and 9 years of age, and you actually followed each child to see how much their cortex changed over that 2 years. And then you look to see how much their phonological processing abilities, that's their sort of ability to do basically phonics or to understand phonemic differences in words. If you looked at their phonological processing improvements and you looked at their fine motor control, she could show that this local thickening effect that she had observed in the what we call the perisylvian region was that the degree to which they had thickening in that region predicted the degree to which their phonological processing improved over that 2-year period. Whereas their improvements in their fine motor control were associated with cortical thinning in the motor areas of the brain. So again, suggesting that these dynamic changes that were going on in the brain structure were related in some way, were associated with individual differences among children in their performance on these different kinds of tasks. And this is one of my favorite studies of children. And this is looking at these fiber tract parameters that I was telling you about. And this is a measure of FA in this fiber tract, the superior corona radiata, showing that the higher FA is in this ascending fiber tract in the brain, the better a child's reading fluency is. But FA in this fiber tract doesn't seem to have any relationship to individual differences in performance on what we call a verbal working memory task. Whereas the FA in this more anteriorly lying tract seems to be related in these same children to differences among them in how good their verbal working memory is, but has no relationship to their reading fluency, suggesting that perhaps there are individual differences in the development of these fiber tracts, not only from one child to the next, but maybe even from one neural system to the next. So we may be getting at something that tells us about the neural basis of individual differences, at least in developing children, in terms of how rapidly they're developing different kinds of cognitive skills. This is some data from my own lab in which we were looking at different kinds of measures, different kinds of tasks. In this case, a measure of response inhibition, how well kids between 7 and 13 years of age, how well they were able to inhibit a response that we had primed in them. So we primed them to make a particular response, and then we, at the last minute, said stop, and they had to stop. It's a complicated paradigm, but take my word for it, that's what it is. And we had another task, which was an ability for the kids to remember where in space in a visual array something occurred and keep that in mind over a delay and then report back the position that something had occurred in a visual array. So these are two different functions that develop quite a lot in children over this age range from 7 to 13. And we found that FA in this fiber tract in the inferior frontal gyrus was related to performance variability on this response inhibition task, whereas FA in this more superiorly lying longitudinal fasciculus tract, that's a part of the neural system that mediates spatial working memory, was related to performance variability in spatial working memory. These findings are not only present in children, they're also present in adults. So here's an example of variability in people's ability to do a spatial visualization task. This is a task that is quite difficult for a lot of people. Me, for example, it's very difficult for me. The person has to be able to kind of mentally rotate an object and figure out which of these boxes is this same box rotated in a different angle. And this is a task that people vary tremendously on in terms of their ability to do it. And this study showed that the measure of microstructure in this fiber tract that's part of the system that has previously been implicated in this function, that the FA in that fiber tract was related to performance variability on this task in adults. So questions have now begun to emerge as these kinds of associations between individual differences in brain morphology or structure of fiber tracts and individual differences in behavioral measures have occurred. People begin to ask the question, could these associations result from experience-related neuroplastic changes in the brain tissues? In other words, could training or experience or practice actually change the brain structure? And is that what underlies these associations? And one of the first studies that raised this question is a now fairly famous study of London taxi drivers, in which the authors studied London taxi drivers, and many of you probably know that London taxi drivers have a really formidable task to learn all the London streets, and it's actually a very demanding job. And what they did was they studied with a technique called voxel-based morphometry, the structure of the hippocampus, which has been implicated in sort of spatial navigation functions, and found that if you compared London taxi drivers to matched controls, there was a difference in the shape of the hippocampus. So London taxi drivers had larger posterior hippocampi and somewhat smaller anterior hippocampi. And of course you could say, well, maybe this just means that people who have a particular hippocampal shape are a little better able to do spatial navigation, so they gravitate naturally to taxi driving as a profession. But what they went further to do was to look to see what the relationship was between the number of months that the taxi drivers had been driving taxis and the different parts of and the size of the different parts of the hippocampus, and they found that indeed the number of months that someone had been a taxi driver predicted this anomaly. So the longer you'd been driving a taxi, the more your hippocampus was shaped in this way, with a larger posterior and a smaller anterior region. And again, this was just circumstantial evidence, but it suggested that maybe it was the actual taxi driving and intensive spatial navigation that was driving this change in the shape of the hippocampus. And since then, a number of other studies have come out that are also kind of suggestive in this way. Here's a study of revealing increased gray matter density in Broca's area. Broca's area is a part of the brain that's been mostly associated with fluent speech, fluent language abilities. But this area appears to be a little bit, the gray matter appears to be a little bit denser in this Broca's area in male symphony orchestra musicians. And this was interesting by itself, but the authors also noted that this area, in the non-musician controls, across the older part of the age range, there was evidence that there was an age-related volume decrease in this part of the brain with in those non-musicians, but there was not any evidence of an age-related volume decrease in the symphony orchestra musicians. And furthermore, if you looked at the younger musicians, it looked like this area became thicker the longer they had been playing in the symphony orchestra. Again, sort of suggesting that maybe what you're seeing is a kind of neuroplastic change, which is actually the result of this intensive activity in this part of the brain associated with their profession, professional music making. And this is probably one of the findings that most directly addresses this issue. This is a study published in Nature in 2004. Normal volunteers came into the lab, and they all were selected for not being jugglers. And half of them they were all scanned with MRI at baseline, and then half of them were taught a simple juggling task. They had to practice until they could sort of do it to criterion. They had to do it competently. And then they were rescanned. And then they were sent away for 3 months and told they were not allowed to do any juggling in the 3 months that they went away, and they were brought back in and scanned again. And the authors were able to show that parts of the intraparietal sulcus, which again have been implicated in this kind of visual spatial function in previous studies, seem to show thickening of the cortex from baseline to post-juggling lessons. And that when these individuals came back 3 months later, it looked like maybe it was moving back in the direction that it was at baseline, the thickness of the cortex in this region. So again, suggestive evidence that there were neuroplastic changes that resulted from this intensive activity occurring in this neural pathway. This is another finding that again suggests that brain morphology is affected by the activity in the brain and other physiological parameters in the brain. And this is a study that was published in 2003 by Colcombe's group, just showing that aerobic fitness that was measured in a group of individuals seemed to reduce the brain tissue loss that occurs across the age range. So this is in their study. This shows the distribution of areas and using one of these computational morphometry techniques, where there was evidence for age-related decline of volume in the gray matter. And this is the distribution of areas that seemed to have a blunt, sort of less decline in those who were most aerobically fit. And you can see it's pretty much the same distribution. So it seems to be a kind of global effect that looks like it might dampen the volume loss that occurs across the age range. This is a study comparing full-term to preterm infants at term equivalent age. So these are preemies compared to full-term babies just at full-term age. And looking at FA, these are the biological development of the fiber tracts in the brain. And these are just the sort of sensory motor fiber tracts that you see here, which are the first ones to myelinate after birth, showing that, interestingly, the preterm infants had higher FA, or what looked like more advanced biological development of these fiber tracts, than the full-term babies did, which was not what most people had predicted from this study, as though perhaps this biological development of the fiber tracts had been accelerated by the extra sensory motor stimulation associated with extrauterine versus intrauterine life. Again, a little bit suggestive that you can change structure with experience and training, stimulation. Now, I've told you a lot of ways that experience, training, and environmental things impinging from the environment, pathogens, drug exposure, and so on, can change the neural tissues. But there's also a lot of evidence emerging about genetic factors that could account for some of the individual differences that we see in brain morphology and brain structural characteristics. And this is kind of coming at it from the other angle. Now the data are pretty strong to suggest that brain morphology exhibits very strong heritability. Twin studies, for example, have revealed that there is very high heritability of brain size, brain shape, cortical surface area, and cortical thickness. And some of the authors of these papers are with us tonight that have really shown the strong, heavy heritability or strong heritability of many brain structural characteristics. And just to give you some idea, here's just some pictures that show you, I don't know, these are just two slices through the brains of two monozygotic twins. These are girls, and they're same-sex siblings. And I think you can see pretty quickly that they look much more similar to each other than they do to their same-sex sibling. And these are dizygotic twins, and they sort of don't look that similar to each other. I mean, I know if you're not used to looking at these, it's maybe hard to see, but these, I think you can see the striking similarity between these two sections that are through the same part of the brain. And this is not surprising. After all, when you look at the faces of identical twins, they're very, very similar to each other, and they don't look that similar to their other siblings. And not only do we see high heritability, but now people are beginning to make observations of the relationship between particular morphological characteristics and specific forms of genetic variation, specific polymorphisms that are present in the genome. And so here's an example of a particular polymorphism in the gene that codes for BDNF, or brain-derived neurotrophic factor, showing that one genotype is associated with smaller volumes of the hippocampus and thinner frontal cortex. This is by Daniel Weinberger's group at the NIMH. Same group, another observation suggesting that the volume, in this case I think of the subgenual cortex and the amygdala, appear to be a little bit different in people with different polymorphisms in this serotonin transporter gene. Here's a really hot off the press observation, which is that this was just presented at the Imaging Genetics meeting yesterday. And it came from several groups here in La Jolla, Anders Dale's morphometry group, Nick Shork's genetics group at Scripps, and a Norwegian group who produced this Norwegian cohort, showing very suggestive evidence. Now, these are data that need to be replicated, because these kind of findings that are obtained from exploratory genetic studies really have to be replicated to be validated. But very interesting observation that people with one genotype show a much less pronounced loss of cortical thickness or reduction of cortical thickness across the age range, whereas people who are carriers of this minor allele, the T allele, show a steeper loss of cortical thickness across the age range. Very interesting observation. Okay. Although the genetic factors appear to guide the development of neural architecture, throughout the lifespan, brain structure exhibits subtle alterations associated with maturation and brain aging. and with dynamic neurobiological responses to environmental factors like drugs or training or experience. I think the picture that we're getting is of neural tissues that are biologically very dynamic. And I think the way we have to think of it is that the tissue biology in the neural tissues at any point in time reflects genetic factors and epigenetic factors and the influence of both recent events and remote events, things that happened a long time ago in the development of the individual. But all of these observations, I think, raise very important questions about the nature of individual differences, behavioral individual differences, for example, and also differences in the course of development and in the course of brain aging. They raise questions about the role of genes and experience and the degree to which behavioral phenotypes really can be shaped by interventions. Future neuroimaging studies will help to reveal these tissue dynamics that are influenced by genes, experience, and exposure to pathogens and drugs, and will help to link them to particular functional outcomes and brain aging outcomes. And the most informative studies are going to employ a whole array of new imaging techniques, and they're going to demand advanced informatics tools for modeling and integrating these complicated data, and they're going to have to be complemented by advanced methods in behavioral science and in genomics research as well. And I'm going to stop here and acknowledge the sources of funding for my research from the NIH and Department of Veterans Affairs, in my Danish research program, the Lundbeck Foundation and the Danish Medical Research Council, my many colleagues at UCSD and VA, the HIV Neurobehavioral Research Center, and also my many colleagues in Denmark where some of the developmental studies have been done. So I'll stop there.